Welcome to another episode of the Globe Program Learning Activity Series brought to you by the Asia Pacific Regional Coordination Office. Before we begin, let me give you a short introduction about chromatography in relation to photosynthesis. Are you ready? Let's begin. Chromatography is one of the procedures commonly used in many fields of science and industry to separate and identify substances within a mixture. A chromatogram is the separation pattern produced by each different mixture. The mixture is placed onto a medium such as paper or chalk, which absorbs water, alcohol or other solvents. As water or alcohol moves up by capillary action on the paper, molecules or substances dissolved in the mixture will travel at different speeds. The water or other solvent moves up the paper because capillary action is stronger than gravity, and capillary action depends on cohesion and adhesion. Cohesion is the mutual attraction of water molecules, and adhesion is the attraction of water molecules to other kinds of molecules, such as raindrops sticking to windows, dewdrops hanging from leaves, wet sheets of plastic becoming stuck to a table, and, in this case, paper. Various colors will appear at certain distances from their starting point because molecules of these colors have different sizes, shapes and solubilities. Molecules that dissolve better in the solvent will move along the paper more easily and quickly and will travel the greatest distance. Other molecules are not able to move as quickly and are left behind. By using paper chromatography, the different colors that make up black ink and pigments in leaves can be separated and made visible. Pigments are colorful compounds that absorb light. Pigment structure and amount determine variations in color. The pigment chlorophyll in leaves helps make photosynthesis happen by absorbing from sunlight the energy needed for putting together carbon dioxide and water to form glucose or food. Chlorophyll gives plants their green color and may hide the other pigments present in leaves. Chlorophyll absorbs all colors of visible light except green, which it reflects to be detected by our eyes. If all colors or wavelengths of visible light are absorbed and none are reflected, the pigment appears black to our eyes. Conversely, if all wavelengths are reflected, the pigment appears white to our eyes. In autumn, changes occur in deciduous plant leaves before they finally fall from the branch. Chlorophyll breaks down and leaves change color when water and sap stop flowing into the leaves. As the green fades, orange color from the pigment carotene and yellow color from the pigment xanthophyll appear. These pigments are also found in foods like carrots, bananas and egg yolks. Carotene and xanthophyll are secondary pigments that support the photosynthetic process by passing their absorbed light energy to chlorophyll. Further chemical changes in the leaves stimulate production of anthocyanin pigments, giving bright red and purple colors. These are also commonly found in plants such as beets, red apples, and purple grapes, and flowers like violets and hyacinths. In the leaves, these pigments are formed in the autumn from trapped glucose. Different mixtures of chlorophyll and other pigments in the leaf give a wide range of autumn colors. Brown colors come from tannin, a bitter waste product. It is important to remember that the key photosynthetic pigment is chlorophyll because the light energy it absorbs is directly used for photosynthesis, while the other pigments have to pass the light energy they absorb to chlorophyll. Okay, time now for some field activity. Are you ready? Let's begin. The following explorations can be conducted by individual students or teams of students. This activity is called investigating leaf pigments. It aims to explore the different color pigments in leaves and understand their importance to photosynthesis. It involves separating color pigments in leaves using a technique called paper chromatography, which can be done individually or in groups. At the end of this activity, students should be able to demonstrate understanding of the following science concepts. In physical science, chemical reactions are everywhere, including in plants. Energy flows through the ecosystems in one direction, and that living systems require continuous input of energy to maintain their chemical and physical organizations. Through this activity, students are also expected to learn the following scientific inquiry abilities: observing and inferring, hypothesizing and predicting, collecting, analyzing and interpreting data, and communicating results and conclusions, designing and developing scientific investigation, using appropriate tools and techniques, developing explanations and predictions using evidence. For this experiment, you need the following materials: White coffee paper filters, paper towels, filter paper or chromatography paper cut to 2 cm wide by 15 cm long. Non-Crayola brand felt tip pens with black, water-soluble ink. Glass or plastic containers, 8 to 10 ounces cups. Water at room temperature. Hot water from faucet. Rubbing alcohol, 70% to 90% isopropyl alcohol. Pencils, popsicle sticks, or drinking straws, scissors. Ruler. Tape. Green leaves and leaves that have changed color from green. Covers for jars, aluminum foil, or plastic wrap. Shallow pan or tray. Globe science log. Hand lens, optional. Mortar and pestle for grinding leaves, optional. Warning: this activity uses alcohol and hot water. Adult supervision is required. Time required for this experiment is 1 class period, or between 45 minutes to 1 hour, for preparation and setting up, plus another 1 class period for observation. Okay, we're almost there. Before starting the exploration, ask students to collect 2 to 3 pieces of large green leaves, and take note of their location and type. Then, ask them to collect 2 to 3 other large pieces of leaves that have changed color, from green. Place each sample in a glass or plastic container, with labels indicating their location and type. In class, using a pair of scissors, cut the coffee filters into strips of 15 centimeters by 2 centimeters. Cut enough strips for the number of leaf samples you have. Next is preparing your pens. Tape a number to each pen, corresponding to the number of leaf samples you have. Then, get 2 to 3 large green leaves and place them in a glass or plastic container. Better yet, put them in a large ceramic bowl such as a mortar, for grounding. Add some isopropyl alcohol, just enough to cover the leaves. Then, grind them using a pestle. Alternatively, you may cut the leaves with a pair of scissors, or tear them up with your hands, and place the cut-up leaves into a glass or clear container. Then add isopropyl alcohol enough to cover the leaves. When that is done, get a spoon or spatula to vigorously stir the leaves and alcohol mixture. Next, pour the mixture into a clear glass container, and cover the container with a lid or plastic wrap. Label your container indicating the name of the leaf, or its location. Then, place the container carefully into a shallow tray containing 1 inch of hot tap water. Keep the container in the tray for at least half hour, or until the mixture has become dark. Add hot tap water if needed. When the mixture has darkened, remove the container from the pan. Then, take one of the coffee strips, and get a pencil, and some tape. Get a small strip of tape, and take the coffee filter strips to the pen, as shown. Next, place the pen with the strip of coffee filter on top of your glass container, containing the leaf pigments. Then, wait between 30 to 90 minutes. If you watch closely, you will see pigments rising from the mixture to the coffee strips. Do the same procedure for the other samples that you have. Then, wait for the pigments to rise up to the coffee filter strips. When this is done, gently remove the coffee strips from the container and from the pencil where it is attached. Then, gently dry it with a paper towel. Next, examine the pigments, taking note of the distances of separation between colors. Finally, back in the classroom, ask students to write and draw their observations, and to share and discuss their findings with the whole group. And finally, get them to discuss the following points. What they think the reason is for the way some colors traveled higher up the strip while others didn't. What can be inferred regarding what pigment molecules are represented on the strip. What can be inferred if green is the dominant color present. Use the Globe Science log sheet to write students' observation. For more advanced discussion points, refer to the GLOBE program activity investigating leaf pigments found at the GLOBE website. For more learning activities like this, visit the GLOBE program website at www.globe.gov. Thank you for watching another episode of the GLOBE program science learning activity. Remember, don't just learn science, do science.